Hey everybody, welcome to Healing Outside the Box. My name is Jean Tiberio and I'm a nutritionist and a wellness coach. This is episode 190 and it's called My Saliva Diagnosed My Disease. Will my skin cure it? But before we get into that, I've got a few interesting articles to pass along to you. The first just came out in the Huffington Post and it's called Everything You Know About Obesity is Wrong. It's a very long article, but I'll give you a link in the show notes if you're interested in reading it. I might also consider doing a podcast episode just highlighting the points brought up in this article, which was very well researched in my opinion. The second article that I'd like to bring to your attention came out in the Boston Globe last Sunday and Monday. It's called Stranger in the House. I have first-hand knowledge of this because I was one of the many people she interviewed for the article. The Boston Globe writer, Linda Machin, spent 2 years researching the current situation with home health care in Massachusetts. Now, these investigations, especially by the Boston Globe, usually happen when things go terribly wrong, and this would be the case here. The problem as she sees it is that there is a lack of oversight and policing in this field. For personal care attendants in Massachusetts, there is no agency, no oversight, and few rules. So people literally have to find a personal care attendant by taking an ad out in Craigslist. But in the end, on the second day, she mentions several people and agencies that are working to make this better. I'll give you a link to that article in the show notes. Even if you don't use personal care attendants or home health aides, it's very interesting. I'll give you a link to this article in the show notes as well. Here's the third thing I want to mention. I also listened to a webinar the other day about the latest news in the diagnosis and treatment of Alzheimer's disease. They basically repeated what we have all been learning lately, and it's this. Alzheimer's disease isn't simply a one-cause disease, and the amyloid plaques in the brain are not the cause. Removing them doesn't fix it. The amyloid plaques that can be seen in CAT scans are more of a symptom of the disease process. They talked about the fact that there are actually different types of Alzheimer's disease. And Alzheimer's disease as a whole is only one type of dementia. The experts list this first type as inflammatory, or the result of some inflammation caused by an invader. Examples they give are like the inflammation resulting from, say, Lyme disease, and, believe it or not, inflammation that might be triggered by a mold. In this type, poor diet is definitely a factor, both with things like pesticides and herbicides and processed food, but also poorly controlled diabetes. Poorly controlled diabetes and heart disease brought on by a poor diet is also triggered by an inflammatory process. Now, the second type of Alzheimer's disease is called atrophic Alzheimer's. This is another way of saying that there's not enough of the good stuff. And one of the good stuff substances may be something like vitamin D. The third type of Alzheimer's disease is toxic Alzheimer's disease. An example of a toxin here might be mercury. No surprise that mercury rots the brain, but we can see now that it's linked to getting Alzheimer's. So this placing of the causes into categories is a good first step in trying to figure out how we might start to treat this disease, and more importantly, perhaps, how to prevent it. So that's an important finding from this Alzheimer's conference that I wanted to pass on to you. Now, back to the subject of this episode. I happen to have a neuromuscular disease that is 100% genetic. They have now found the defective gene, and they know the exact protein that this gene codes for. Back in January of 2017, I was involved in a clinical trial for an experimental drug to treat my specific disease. This drug is a precursor of the missing muscle protein, and it's given in pill form. Months later, my hopes died on the vine because the clinical trial was stopped in phase 3. Since then, I've spent a lot of time researching this disease on the internet and similar genetic diseases on the internet because, as I keep trying to tell myself over and over again, I'm not a quitter. And no, I don't have the answer to the question I am posing, namely, will it be possible in the future to fix my defective DNA and put it back into my body? So my short answer is I don't know. But because I have a classic genetic disease and therefore skin in the game, and my defective gene has been found, I decided recently to take a look around and see what's up on the internet in genetic therapies. It may be by thinking out loud I can put some thoughts into your heads about this whole idea in general, because if it's possible to fix genetic diseases by gene therapy, it could affect so many people around the world in so many ways. So here's my story. I was first diagnosed with muscular dystrophy, unknown type, back in 1982. At that time, they were starting to type the muscular dystrophies, but only by appearance of symptoms. In other words, if your shoulder muscles had weakened first, you might be told that you have a certain type of muscular dystrophy, versus your muscles below the knee. Then you might be typed with another form of muscular dystrophy. But it was only a guess. Muscular dystrophy as a term basically stands for weak and wasting muscles. The type with the most research is Duchenne muscular dystrophy. And Duchenne is best known because it's the most common and because only boys get the disease. So that told the scientists what chromosome to look for. So your gender is determined on your sex chromosome. If you're XX, you're a girl, and if you're XY, you're a boy. With Duchenne muscular dystrophy, if you're an XX or a girl, you may be a carrier for Duchenne's but don't have the disease. If you're XY, then 100% of the boys, if their mother is a carrier, have the disease. And that's about all they knew of the most common form of muscular dystrophy at the time. Then, back in 2016, when I was visiting my neurologist, I was told that they were testing for a very rare neuromuscular disease. It was only a simple DNA saliva test, so I thought, why not? My DNA test came back positive for this rare disease, and it's called hereditary inclusion body myopathy. It's also called GNE myopathy, for obvious reasons. It's easier to say. I was told at the time that it is literally a 1 in a million chance that you'll get this disease, and it is more often than not seen in Iranian Jews. That's interesting, I thought, and perhaps because I am an Italian-Irish Catholic. So what's this disease all about? Well, again, because it's purely genetic, everyone is born with it. The symptoms usually get noticed with a foot drop. In other words, you can't pick up your toe when you're walking, and then you trip on your feet and fall forward. That was happening to me like crazy. But when it was happening on a regular basis, and I kept going back to the doctor, it was actually a full year before I was diagnosed with muscular dystrophy. There's a type of muscular dystrophy that results in foot drop, and that's the type that they thought I had for years. Now, when I looked into this hereditary inclusion body myopathy, I found a few interesting things. This disease can progress from foot drop to bedbound in either 2 years or 20 years. So there's a lot of variability in the progression, as the scientists would say. Now, I've been in a wheelchair since 1990, and right now I can only move the fingers of my left hand and turn my head from side to side a little. But I'm definitely not bedbound. And I'm a podcaster, for goodness sake. And remember, the foot drop was happening in 1981. So personally, I've learned that predicting what might happen in the future is a guessing game. Anyway, the GNE gene that is defective in my case sits on chromosome 9. This gene codes for a protein that is called sialic acid. This is one of a bunch of surface proteins that sit on the cell membrane or the outer wall of the muscle cell. So we know the defective gene, the protein, where it is, and basically what it does. Why can't we stick it back in there, for goodness sake? Well, it turns out that it's more complicated than that. Although it appears that this sialic acid protein may be involved in energy metabolism, it seems to have other roles as well, and it interacts with other proteins and other pathways. Here's the thing about this. Because it's a protein, our bodies simply can't swallow it as a pill, because our bodies think it's food, and it will break it down into little amino acids to be used for other purposes. That may be part of the problem with that clinical trial that I was involved in January of 2017. This took place at NYU on the 20th floor. I have a story about that in one of my podcast episodes, and I'll see if I can find it to link it for you. They were giving everybody in the study a protein that was a precursor of sialic acid, and they were giving it in a very large dose. It was an extended release form of the drug, so they were hoping that some of it would remain intact and get into my bloodstream. Now, as I mentioned in the story in a previous episode, I definitely found some improvement with a physical therapy program that I put myself through. I wasn't supposed to be doing physical therapy, but I did anyway, and I just fibbed about it. So much for clinical studies being completely accurate all the time. But according to the neurologist in the study, there was not enough improvement for the drug company to continue making this expensive drug for a rare disease. In other words, not enough profit for a 1 in a million disease. Although I personally think, having done a few studies in the past when I was young, that if the mode of therapy works, it could potentially be used for any genetic disease where we know the defective gene. So let's talk about potential therapies that I've come across. One of the potential therapies is called gene editing. Now, if you were watching 60 Minutes when they had a story on CRISPR, or the CRISPR gene therapy, that's basically what we're talking about here. I'll give you a link in the show notes, because the CBS News website keeps it up for people to still see. Now, gene editing in general means changing the DNA of a person inside the person, rather in a test tube. Ideally, this can be used to change a mutation back to the normal sequence. By the way, CRISPR is without an E, and it stands for Clustered, Regularly Interspaced, Short, Palindromic Repeats. Now, this CRISPR is attached to a protein. These CRISPR substances were originally found in bacteria and function as a defense mechanism against attacks from viruses. And yes, you heard me. Viruses attack bacteria. They use this CRISPR thing as scissors to snip out the mutated patch of DNA. So the faulty DNA patch is cut out and a normal DNA patch to make the normal protein is glued back in. If it sounds more difficult than a simple cut and paste, you would be correct. But scientists that are working on this are surprisingly optimistic. Okay, that was CRISPR therapy. Now let's talk about straight stem cell therapy. Do you remember Dr. Oz talking about this 20 years ago on Oprah? I do, since again I have skin in the game. So where is this in 2018? Now, stem cell therapy is based on the theory that all of the different cells in our body started out as stem cells, and these cells multiply by division and then differentiate into a specific kidney cell or blood cell or muscle cell. So how do we make stem cells into muscle cells? Again, easier said than done. We have learned valuable information since the Dr. Oz days. We now know that all of our cells carry stem cells within them. And I'm sure you've heard that now a person's skin cells can be spun down and made into their stem cells in the lab. Unfortunately, the 8-year halt on all stem cell therapy during the George W. Bush years slowed down this whole process. I guess terrified people just ran in circles, afraid their baby would be stolen and carved up for stem cells. But now it's 2018, and theoretically right now, you can use your own skin cells to produce your own stem cells and make them differentiate into kidney or liver cells, or in my case, muscle cells. They would not be rejected because we are the original host. So what's the problem with that? The first hurdle is to make sure that these cells can reproduce in enormous numbers without forming tumor cells. That's the whole first do no harm thing, which personally I find annoying. If I know I'm slowly dying of an incurable, debilitating disease, I should have the choice to sign a piece of paper saying I'm okay with a 0.1% chance of getting tumor cells, which I would be. Now, the second hurdle is the source of the stem cells. Bone marrow, as we know, can be used as a source for stem cells, but the number of stem cells that can be derived from that are presently in small numbers and not large enough to make it a viable source for everyone. So this guy from Japan named Yamanaka turned a small amount of skin tissue into large numbers of stem cells in the lab, and he won the Nobel Prize in medicine for that. So we can make stem cells in the lab. The next hurdle is making a huge quantity of them while retaining the quality so as not to accidentally make tumor cells. Recently, there were some scientists at the University of Wisconsin that are starting to make large-scale amounts of stem cells. And basically, they're saying that the only way to bring the price of stem cell therapies down to make it affordable is to make consistent, high-quality, large amounts of stem cells that will be able to differentiate into all these different tissues. I'll give you a link in the show notes to that scientific report. It's very interesting. If you think about that, this could potentially help people with injuries heal much faster. Theoretically, T cells can be generated this way and help people with HIV. The examples are potentially unlimited. Another research group at the University of California has isolated human muscle cells and shown that the stem cells inside them, this is inside the developed muscle cell, might be able to be used for therapy with both injuries and something like my disease. This made me do a happy dance, as Phil Rosenthal likes to say on Somebody Feed Phil. By the way, if you haven't seen that show, you must see that show. If you want a little pep me up and don't have Red Bull lying around, it will make you laugh. I'll give you a link to the University of California study in the show notes as well. Now, some smart guy somewhere has figured out how to convert fat cells to stem cells instead of using bone marrow. As we know, extracting bone marrow is very painful and shoots up the risk of infection. Fat cells, not so much. Technically, a surgeon could scoop out some fat cells, hopefully in the belly, I'm just saying, and convert them to stem cells. And then theoretically, they could make some new muscle, blood cells, or even T cells. One of the first approvals for marketing a stem cell product in Europe has recently been secured by a Nobel laureate-led company in Wales. This approval is related to curing heart failure. The European Medicines Agency, called EMA, wants to test these procedures as part of a drive to evaluate potentially life-saving treatments more quickly than in the past. I will give you a link to that source in the show notes as well. So this is where I am. After being discouraged over and over when I thought there was a cure for my disease on the horizon, then my hopes were dashed. I thought there was a cure, baby, yippee, and then my hopes were dashed. But right now, I feel more optimistic that stem cells in some way could be what cures my disease. Now, that's all I have for you today. Be well and stay tuned next week for another episode of Healing Outside the Box.